This is the DNA aptamer COVID-19 antigen rapid test kit. We have 3 test tubes labeled T1, T2, and T3. We will use T1 as the testing tube, while the cap of T2 and T3 will act as the catalyst for the testing process. We also have the saliva swab, testing strip, and a saliva collection bag. To start, we recommend video recording the entire testing process for analysis later on. Take test tube T1 and shake vigorously to mix the solutions inside. Remove the cap. For convenience, we will tear open the Ziploc bag to collect the saliva sample. Retrieve a small sample of saliva with the saliva swab, and gently rotate inside the sample tube at the very top. Be sure not to make contact with the solution in the tube. Replace the T2 cap onto the sample tube. Set timer for 3 minutes and consistently mix the solution in the test tube for about 30 to 40 seconds. Shake once vigorously to allow the solution to settle together at the base of the tube and leave for the remainder of the 3 minutes. Remove the T2 cap and replace with the T3 cap onto the sample tube. Set timer for 2 minutes and consistently mix the solution in the test tube for about 30 to 40 seconds. Shake once vigorously to allow the solution to settle together at the base of the tube and leave for the remainder of the 2 minutes. Remove the T3 cap. Set timer for 2 minutes and place the test strip with the U for up into the solution tube. Start the 2 minutes count. by comparing the result of this test trial with the following reference images. We can see that the trial has tested negative. In the healthy reference results that tested negative for the presence of any antigen, we can see that the dark brown lines exhibit a symmetrical pattern in both the length and the position of the upper and lower portions. However, when the dark brown lines exhibit any type of asymmetrical patterns, such as differences in length, non-centered lines, or significant edge lines, then this indicates the presence of some type of antigen, and the patient should be further examined by a doctor. In the case of significant asymmetrical coverage by the dark brown marks in the lower portion of the test strip, this is an indication of the presence of coronavirus.